So, let's go. Right now, across 5 different worlds in our solar system and beyond, there are chemicals, patterns, and reactions that science cannot fully explain without invoking life. Not alien civilizations, not little green men, molecules, mineral patterns, gas that shouldn't exist. Things that keep surviving every attempt to explain them away. And NASA built a 7-level scorecard to track exactly how close each discovery gets to the real thing. They call it the confidence of life detection scale. Level 1 is, that's weird. Level 7 is confirmed alien life. Nothing we've ever found is scored above a 3. This is a tour of everything on that scoreboard, weakest to strongest. And the strongest one is sitting in a tube on Mars right now, waiting for a ride home that might never come. Number 1. The exoplanet that smells like garlic. 124 light years away, there's a planet called K2-18b. It might have a liquid ocean under a thick hydrogen sky. It might be a ball of gas with no surface at all. We genuinely don't know. In 2025, a team using the James Webb Space Telescope announced they'd found traces of dimethyl sulfide in its atmosphere. On Earth, that molecule is made almost exclusively by marine phytoplankton. It's the smell of the ocean. Its chemical cousin, dimethyl disulfide, smells like garlic and rot. Both were detected at 3 sigma confidence, which sounds solid until you know the gold standard for discovery is 5 sigma. And then the rebuttals started arriving. Multiple independent teams reanalyzed the same data. 87.5% of their analyses found no evidence for either molecule. NASA quietly declined to issue a press release. And in 2024, dimethyl sulfide was found on a cold, dead comet, meaning it doesn't require biology at all. The concentrations didn't help either. On Earth, dimethyl sulfide exists at less than 1 part per billion. On K2-18b, it's over 10 parts per million. Thousands of times stronger. Either this planet has the most wildly productive biosphere ever conceived, or something non-biological is making the molecule, and we just don't understand the chemistry yet. The lead researcher himself admitted that if the planet has a deep atmosphere, no biology is required to explain the signal. Cold scale score, roughly 1, barely on the board. But that's not a failure. That's what honest science looks like when a telescope is pointed at something 124 light years away, and the worlds that score higher are a lot closer to home. Number 2. The planet that smells like death. Venus will kill you in about 15 different ways. The surface sits at 460 degrees Celsius under 90. Atmospheres of pressure, hot enough to melt lead, crushing enough to flatten a submarine. But between 48 and 60 kilometers up, floating in the cloud deck, something strange is happening. Temperatures up there run between 0 and 60 degrees. Pressure is about 1 atmosphere. It's the most Earth-like environment in the solar system outside Earth. And in September 2020, an astronomer named Jane Greaves pointed a telescope at those clouds and found phosphine, a molecule that, on Earth, is made almost exclusively by living things. Anaerobic bacteria, penguin stomachs, that's about it. The amount she detected was about 20 parts per billion. For context, Earth's atmosphere has a few parts per trillion. Venus had thousands of times more. Then the scientific community turned on the result like it owed them money. Data processing errors. The signal might be sulfur dioxide, not phosphine. The telescope calibration was wrong. Team after team took shots. The number got revised down to 1 to 4 parts per billion. Some groups found nothing at all, but the original team didn't fold. A researcher named Dave Clements led a follow-up campaign that collected 140 times more data than the original detection. He found phosphine again. He also found tentative signs of ammonia, another molecule that has no business being in clouds made of sulfuric acid unless something is actively producing it. And then he found the pattern that changed everything. Every single observation that detected phosphine was taken when the Venusian atmosphere was rotating from night into day. Every observation that found nothing was taken during the day-to-night transition. Ultraviolet sunlight destroys phosphine during daytime. It gets baked out. That's why some telescopes saw it and some didn't. They were looking at different times. In September 2024, preliminary data from a new observation campaign suggested concentrations of 300 parts per billion at 55 kilometers altitude, right inside the habitable cloud layer. If that number holds up, it's enormous. Meanwhile, Data from a 1978 NASA probe that visited Venus was reanalyzed by a completely separate team. They independently found phosphine signatures inside the clouds. Their analyses, they say, remain unchallenged in the scientific literature. A comprehensive review of every known abiotic way to produce phosphine on a rocky planet, volcanism, lightning, meteorites, photochemistry, surface reactions, concluded that none of them can produce the quantities being detected. Something is making phosphine in the clouds of Venus. We just don't know what. A NASA probe called Da Vinci will descend through those clouds later this decade, carrying instruments that can settle the question directly. Until then, this molecule keeps surviving every attempt to kill it. Co-LD scale score, roughly 2. The detection is held up. All of the source is a complete mystery. And the next world on the list isn't hiding its secrets at all. It's throwing them at us. Number 3. The moon that hands you free samples. Enceladus is 313 miles across. That's roughly the distance from Los Angeles to San Francisco. A moon so small it would fit inside the state of Arizona. And it is screaming. 4 enormous cracks near its south pole, nicknamed the Tiger Stripes, are blasting ocean water into space at 800 miles per hour. 5 times faster than a Category 5 hurricane. Warm, salty water from a global subsurface ocean, erupting through miles of ice and spraying into the vacuum of space like a fire hydrant, nobody can turn off. Between 2004 and 2017, NASA's Cassini spacecraft flew through those plumes and tasted what came out. Salt water, complex organic molecules, molecular hydrogen, which on Earth is food for microbes at the base of the tree of life, silica nanoparticles so small they're measured in billionths of a meter, and the only way to make particles like that is in salty water heated to at least 90 degrees Celsius. That means hydrothermal vents on the ocean floor of a moon of Saturn. In 2023, scientists found phosphorus locked inside the ice grains shooting out of those cracks. Concentrations 100 times higher than Earth's oceans. First time phosphorus had ever been detected in an ocean off Earth. That completed the checklist. Carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. Every element life needs. All 6. No other body in the solar system checks every single box the way Enceladus does. One of the mission scientists put it simply. It would be like a candy store for microbes. Here's the thing, though. Cassini wasn't designed to find life. When it launched, nobody even knew the plumes existed. It stumbled into one of the most important discoveries in the history of space exploration, with instruments built to study ring particles and magnetic fields. And there's a catch that makes it worse. Research from 2025 suggests the ocean is probably stratified, layered like oil and water in a jar. Biosignatures from the deep ocean might take hundreds of thousands of years to drift up to the plumes. By the time they get ejected into space, they might be unrecognizable. ESA is studying a dedicated Enceladus mission for the 2040s. Fly through the jets, land on the surface, scoop up samples. But right now, this tiny frozen moon is spraying the contents of its ocean directly into space, handing us free samples of a world that has everything life needs, and we don't have a single spacecraft on the way to catch them. Co-LD scale score, roughly 2, not because the evidence is weak, because we haven't built the right instrument yet. Number 4, the experiment they buried. July 20th, 1976, Viking 1 touches down on Mars. 2 months later, Viking 2 lands 4,000 miles away. That's roughly the distance from New York to Hawaii. Both of them are carrying biology experiments. The one that matters is called Labeled Release, designed by a scientist named Gilbert Levin. Simple concept: take a sample of Martian soil. Add a drop of nutrient broth tagged with radioactive carbon-14. If microbes eat the nutrients, they'll exhale radioactive gas. Watch the air above the soil for the signal. The signal appeared at both sites, 4,000 miles apart. Radioactive gas rising from the soil exactly the way metabolism looks on Earth. When they heated the soil to sterilize it, the response stopped. When they heated it to just 50 degrees, low enough that most chemicals wouldn't care, but high enough to stress cold-adapted organisms, the response weakened. Every control behaved exactly the way biology predicts. But another instrument, a gas chromatograph mass spectrometer, found no organic molecules in the soil. And that killed it. If there was life, where were the dead bodies? The scientific consensus snapped shut. Must be some unknown chemical oxidant. Case closed. Move on. No follow-up metabolic experiment has been sent to Mars in 50 years. Levine spent the rest of his life arguing his experiment worked. He was marginalized, dismissed, called a crackpot. His co-experimenter, Patricia Strott, initially disagreed with him. But by the late 90s, as evidence for water on Mars kept accumulating, she changed her mind. Then, in 2008, NASA's Phoenix lander discovered something in the Martian soil that changed the entire argument: perchlorates at 0.5% concentration. Perchlorates decompose organic molecules when you heat them. The gas chromatograph that found no organics worked by heating the soil. It may have been destroying the very evidence it was looking for. Levin died in 2021 at 97 years old. He never got NASA to fly an improved version of his experiment. One chemist who reviewed the full body of evidence said it plainly. Gill was marginalized. He was regarded as a crackpot by the community, even though he's no crackpot. And at the end of the day, he's probably going to be proven right. Now here's where the hard science ends and speculation begins. A team has since proposed a model for what Martian microbes might actually look like. Photosynthetic bacteria that store oxygen during the day and go dormant at night. They call the model Barsoom, after the name for Mars in old science fiction. It's a hypothesis, not a finding. But the labeled release results are real. The perchlorate discovery is real. And the 50-year silence that followed is real. Cole-D scale score, it scored a 1 or 2 in 1976. The perchlorate discovery retroactively moved the goalposts. Depending on who you ask, it's still sitting right there. And the reason that matters is because of what's happening on Mars right now. Number 5, the leopard spots, July 21st, 2024. Perseverance is rolling through a dry riverbed in Jezero crater when it stops in front of a rock, partially buried in the ancient channel floor. Arrow-shaped, about the size of a coffee table, they nickname it Chayava Falls. The rock is covered in spots, millimeter-scale concentric rings, dark and light, scattered across the surface like a leopard print. The team calls in every instrument the rover has. Pixel fires X-rays. Sherlock fires a deep ultraviolet laser. They map every chemical signature down to fractions of a millimeter. The leopard spots contain vivianite and graphite. The iron phosphate and iron sulfide locked together in concentric patterns with organic carbon threaded through them. On Earth, this specific mineral combination, in this specific arrangement, forms overwhelmingly through one process. Microbial metabolism, microbes eating organic matter and using iron minerals as their energy source, leaving behind these exact chemical fingerprints in these exact patterns. The rover also found the rock is made of clay and silt mudstone. On Earth, that's the single best material for preserving microbial fossils. Perseverance drilled a core sample and named it Sapphire Canyon. For a year, the science team tried to explain the leopard spots without biology. They proposed every abiotic mechanism they could think of. Slow chemical diffusion, geological redox reactions, none of them fit the full picture. In September 2025, they published their findings in Nature. The peer-reviewed conclusion, no strong evidence for any non-biological explanation they studied. This is the first single location on Mars to show all three of life's ingredients together, liquid water, organic molecules, and an energy source, in one rock. The acting NASA administrator said it directly. This very well could be the clearest sign of life that we've ever found on Mars. The project scientists put it differently. We basically threw the entire rover science payload at this rock. We're pretty close to the limits of what the rover can do. And that's the problem. The sample is sitting inside a sealed tube, locked in Perseverance's belly. The rover is healthy. It can operate until at least 2031. But it can't analyze Sapphire Canyon the way an Earth laboratory could. It can't put it under an electron microscope, can't run isotopic analysis, can't do the one thing that would settle the question. Mars. Sample return, the mission designed to bring these tubes home, is in limbo. Co-LD scale score, 2 to 3, potential biosignature identified, contamination ruled out, abiotic alternatives studied and found lacking. But not fully eliminated, not without getting that tube to a lab. If those samples come back and a terrestrial laboratory confirms biology, the Coil D scale hits 7. That's the end of the question. That's the answer. If they never come back, we might never know. The answer to the biggest question our species has ever asked might already be collected. Drilled out of a leopard-spotted rock in a dry Martian riverbed, sealed in a tube locked inside a robot that's still driving, waiting for a ride home,